In traditional Mendelian genetics, genes sort independently. If you think of gametes as train cars, the alleles are like independent passengers. A and B can freely choose whichever separate car they want to board. But what happens when two genetic traits are located right next to each other on the exact same chromosome? Our passengers, A and B, are suddenly physically shackled together inside a single car. They can no longer move independently. This physical constraint breaks Mendel's rule, forcing these genes to travel together as a single unit, a phenomenon known as genetic linkage. The biologist who proved this physical reality was Thomas Hunt Morgan in 1910. Through his experiments with fruit flies, Morgan demonstrated that genes occupy specific physical addresses on a chromosome, grounding Mendel's abstract mathematics in physical reality. For your WBCHSE board exam, you need to know this exact definition. Linkage is the tendency of genes present on the same chromosome to remain together and transfer as a single unit to the next generation. Because linkage restricts genetic variability, it deviates from classic Mendelian expectations. If genes are linked, you will not get the standard 9:3 to 3:1 phenotypic ratio. To solve linkage problems, we have to change how we write our genotypes. The standard Mendelian notation, writing it straight across as capital A, capital B, lowercase b, fails to show us where the alleles physically sit on the chromosomes. Instead, we use the vertical bar or slash notation, first observed by Bateson and Punnett as coupling and repulsion. These two vertical bars literally represent a pair of homologous chromosomes. We plot specific alleles on each side to show exactly what's sitting on the left homolog and what's on the right homolog. To successfully solve a linkage problem on your test, you must map the physical reality of the alleles. You have to know which alleles share the exact same chromosome. Let's look at the first common arrangement on this chromosome map, using maze traits, C for color and S for shape. Here, both dominant alleles are grouped together. This is called the cis or coupling arrangement. Cis means same side. It occurs in a dihybrid when both dominant alleles are physically located on one chromosome and both recessives sit on the other. When this parent forms gametes, the genes stay linked. That means the intact parental gametes, big C big S and little c little s, will be produced in high frequency, making up over 50% of the total gametes. Crossing over can sometimes break this physical link during meiosis, but it is relatively rare. So the recombinant gametes, the mixed-up big C little s and little c big S, will always represent a minority of the offspring. There is a second possibility. This is the trans or repulsion arrangement. Here, a dominant allele is paired with a recessive allele on the exact same chromosome. Pause the video right here for 5 seconds. Based on this physical arrangement, try to predict which two gametes will be the high-frequency parentals and which will be the rare recombinants. Let's check your work. In the trans arrangement, the high-frequency parental gametes are big C little s and little c big S. The all-dominant big C big S combination is actually the rare recombinant here. The term parental does not automatically equate to dominant. It refers strictly to how the alleles were originally physically arranged on the parent's starting chromosomes. This side-by-side comparison shows you that identical dihybrid genotypes can yield completely different primary gametes, based entirely on their physical arrangement. Exam cheat sheet. 1. Linkage is the physical association of genes, breaking Mendel's independent assortment. 2. Always map phases. Cis yields AB and AB high-frequency gametes. Trans yields AB and AB. 3. Linkage strength is inversely proportional to distance. Sketching vertical bars prevents errors.